organic chemistry in in biology and drug development. Okay. So, we have discussed so far the first part of this course, that is the organic chemistry in biology, and we have now the perfect background to go into the second topic, that is the drug development process and the role of organic chemistry. How does it help? Okay. Now, before we go on to the actual organic chemistry, we need to know certain aspects of this drug discovery process. First of all, what are drugs, and then what is the safety margin or all chemicals which affect which has some effect on the body, whether all are drugs or not. So, that is I think some preliminary knowledge is required before we move on to the actual drug design and discovery process. Now, drugs are basically compounds that interact with a biological system to produce a biological response. So, basically if you have a living system and you take a chemical and that chemical goes inside and produces some effect, that is called the biological response. Now, the question is how safe are these chemicals which produce some biological effect. So, this definition basically includes any chemical which produces some biological effect. Now, that biological effect may be a toxic effect, may be having a lethal action on the living system, or may be having a beneficial effect. Okay. So, our definition obviously that drugs should be such chemicals which have beneficial effect on a living system like the human, okay, because we will consider the human as our model. Now, no drug is totally safe, that we most of us know that all drugs have some side effects, and the quality of drug varies according to the side effects that they might have. Okay. Now, to minimize the side effect and to maximize the biological response, what we need is to adjust the dose level, what is called a dose. Like some pills are taken in 500 milligram, and maybe the restriction is that you can go up to 4 times a day, some could be 2 times a day. Now, how was this dose determined? That is basically determined by the beneficial effect vis-a-vis the toxic effect that it might have. Okay. So, if you cross certain dose, the same drug may act as a poison, can produce a toxic effect or sometimes a lethal effect, that the person may die of that. Okay. So, dose is very important, and to begin with, that no drug is totally safe, and what are drugs I already told you. Now, this dose development, that how to maintain the proper dose, because is very important, because as I told you that a drug can be a medicine or it can be a poison. Okay. The therapeutic index is a parameter which tells you how safe is the drug. That means, suppose if it is found that a 500 milligram dose is sufficient to produce a beneficial effect, and while suppose if you cross 10 gram, a 10 gram dose causes a toxic effect. Okay. So, that means you have a therapeutic window, that 500 milligram is the beneficial effect and 10 gram is the toxic effect. So, in between you do not have any problem. Okay. So, you can take 500, even you can take 1 gram, so long as you are below the toxic dose. So, that is beneficial to the person or to the human. Now, if this window is very small, the beneficial effect and the toxic effect, then there is a problem. And you know, the drugs today has been it is a misnomer in the context of in today's context, because we are hearing drug addiction, drug addiction and then drug poisoning, all these things are there. So, basically what we are talking about, drugs which are having beneficial effect, and that can be regarded as a medicine. So, the drugs which are having beneficial effect are medicines, and drugs, so all medicines are not drugs. The drugs that we talk about in relation to addiction is they are basically create some kind of addiction, so that the person takes the medicine, even if takes the drug, even if he or she does not require that, but his mentality is such that he thinks that taking that particular chemical as a drug will cure whatever problem he is having. Okay. Now, there we have heard that many cases of death of drug poisoning, and that is because these people cross the therapeutic index of the whatever is prescribed to have a beneficial effect. So, once they cross the therapeutic index, then or the toxic dose, then they are going to have this toxicity related deaths. Okay. Now, it is very interesting that the body, the human body is made up of a very complex biological system. It has got a very complex biological system. So, what will happen now, if you take a chemical, obviously they are going to interact with this complex system. That is nothing abnormal, that any chemical if I take, that is going to affect the chemistry or biochemistry that is going on in the body. That means, on the just in general the metabolism of the body. Now, if these complex processes are all being targeted or are being disturbed by this external agent, which we call the drug, then that is not a very safe drug, or that is not a very specific drug. If a chemical interacts with several biochemical agents or biochemical processes inside a living organism, then it is not a good drug. Okay. Now, that means what we have, a good drug is good when it basically it targets mostly some of the biological agent that is present inside the body. It could be a protein, it could be nucleic acid, it could be carbohydrate, whatever it is, or lipid, okay, and without causing any toxic or unwanted side effect. But as I told you, no drug, there is no drug in the market that is that completely satisfy this condition. That means, that only targets only one particular molecule inside the body, and then without having any toxic or other side effects. Now, side effects come from interaction of this chemical or the so called drug with other molecules which are not targeted in principle. Okay. Suppose I have some cholesterol related disease, hypercholesterol. Okay. The cholesterol is higher than what is expected in the normal human being. Now, if cholesterol is very high, that means the cholesterol is made in the body at a higher level than what is required. Now, who makes the cholesterol? It is the enzymes that are present in the body. Okay. So, these enzymes, suppose I target one of the enzyme which are involved in the biosynthesis of cholesterol. So, if I inhibit that enzyme, then the cholesterol biosynthesis will stop, or it will be modulated, because depending on the type of inhibition and the IC50 values of the inhibitor. But if that inhibitor, what is supposed to reduce the cholesterol level, now suppose that interacts with other some liver enzymes which are essential for metabolism of our of many of the chemicals, or it is targeting to other enzymes which are involved in metabolism of the diet, then what will happen? Then I have the side effect, or this chemical produces acidity inside the stomach, like what aspirin does. Okay. Then that is not a the more the side effect, that means more the interaction with other chemicals inside the body other than the target, then you have this toxic effect arising from the drug. Okay. But just to be very clear that no chemical, no drug or no medicine in the pharmaceutical market today is that completely satisfy these conditions, that it should not have any side effect, no toxic effect, and its therapeutic window should be very high. So, that is no drug satisfies this, but some come very close to these ideal drugs. One example is penicillin. Penicillin has been one of the safest and most effective antibacterial agents ever discovered. Okay. It is now why it does not have, that means it is not targeting other enzymes or nucleic acids or any other biological molecules that are essential for the human being. It is targeting only towards the bacteria that is causing the disease. And when we discuss the mechanism of action of penicillin, you will definitely know that why it is so specific and why it is so effective and why it does not have side effects. However, it is still, it has got some problem, like many of the individuals can experience severe allergic reactions to this compound. So, whenever you go to the doctor and if he is prescribing a penicillin, the first thing that he will ask is that are you allergic to sulfur, because penicillin has a sulfur moiety. So, then he cannot, if it is allergic, if the patient is allergic, then he will not be prescribed any penicillin type molecules. So, other types of antibacterial agents has to be prescribed. Okay. Now, this, so penicillin is relatively safe, because if you do not have allergic reactions, then it does not have much other side effect. There are some other drugs that are distinctly dangerous. Morphine is one such example. Morphine, you know, that is a natural product and that is used as an analgesic, that means pain reliever, and it is given mostly to terminally ill patients suffering from cancer, because the pain is too much and at that time one has to take morphine. However, the problem is its side effects, it has got side effects, and one of them is what is called tolerance. That means, if today 500 milligram of morphine is sufficient to give me relief from the pain, but after 1 month 500 milligram may not be sufficient. I have to increase the dose. The same thing happens with many of the sleeping tablets, that you start with a very low dose, but as days pass by, that the dose has to be increased. Now, this is what is called tolerance. That means, if the body develops tolerance very quickly, that means the morphine dose has to be increased. And so, basically you run the risk of crossing the lethal dose, the dose that is required to kill the person, that is that can be achieved, or sometimes that may happen. Similar is the case of heroin, that is another derivative of morphine, which is also a very good pain reliever, but it is not prescribed, and morphine is also, these are only available on prescription. It is not to be used by anybody without any prescription, because of this addiction problem and the problem of tolerance. Okay. Now, the next question is, what are these? I already told about this, that drugs are mere chemicals, but they are entering a world of chemical reactions. That means, they are entering a biological system, which is called a biological world. and with which it will interact. Therefore, there should not be nothing odd in the fact that they can have an effect. However, the surprising thing is that they can have specific effects. These chemicals that can be branded as drugs when it only have some specific effects. That means, it mostly interacts with something which is the causative agent of the disease, but without interacting with others. But as I told you, side effects are there, but the medicinal chemists or the persons who are associated with drug discovery process, they try to minimize this side effect while maximize the efficacy of the molecule. Efficacy means that efficacy towards relief of the disease. Now, the question is, what are these targets? I am telling you a lot of time that it could be, the targets could be proteins. Now, proteins can be enzymes. So, you can target enzymes, you can target receptors. Receptors are basically proteins which are on the surface of a cell, and where a small molecule binds and then it produces some effect, and which is very important. And so, some may be overactive receptors. So, you have to make a molecule which goes and binds and stop the signaling pathway or reduce the extent of signaling pathway, and some could be transport proteins. That means, proteins which transport molecules inside the cell. So, if you want to lower the metabolic activity, then you have to target these transport proteins, because transport proteins are basically, they are targeting they are taking important molecules from outside and taking it into the cell. So, that you have to for many diseases that needs to be stopped. So, these are the proteins, that means, they are mainly enzymes, receptors, transport proteins. So, they are the good targets. The nucleic acids, DNA and RNA. Now, nucleic acids are targets mainly in case of when you want to kill the cell by a molecule which goes and destroys the nucleic acid, and this is typically what is required in case of treatment of diseases like cancer, because where you want to destroy the cell, the cancerous cell. So, you have to be most of the drugs that are based on nucleic acids, but not all that targets the nucleic acids are anti-cancer agents. They could be antiviral and not antiviral, they could be your other degenerative processes, they are involved. And apart from these nucleic acids, but again just nucleic acids are mostly targeted in case of cancer, and for other diseases we generally target the proteins, and amongst the proteins I said enzymes, receptors and transport proteins. The targets, one interesting point is that the drugs we take are really small molecules. On the other hand, they are interacting with molecules, which are very big, like these proteins are much bigger as compared to the small molecule that we use as a drug. I am sure you have never noticed a drug which has got a molecular of say 10,000. It is always a small molecule, and this small molecule interacts with a very large molecule. Okay. Now, so when it interacts, we have gone through the enzyme chemistry, that the enzyme also takes up small molecules and converts it into products, and there the molecules are very small and they go and bind to what is called an active site. Now, enzymes after binding the small molecules, after binding to the enzymatic site, they undergo some transformation. Okay. Now, there are proteins which has got a binding site, but there is no as such no chemical reaction. But what it does, as it binds, it creates a disturbance in the on the enzyme, and that disturbance creates a change in conformation, and the change in conformation creates a signal, and that signal is then transmitted into the cell or other cells, and then other biochemical processes which are very important for the survival of the cell, that happens. Okay. So, basically we can say that usually the targets are macromolecular targets, proteins or nucleic acids, and usually they have a binding site, especially if they are proteins. They have a site which is called a binding site, and we know that what is the active site. Usually it is a, if this is a protein, so usually what they have is a binding site like this. So, the small molecule goes here and binds, okay, and then creates, either if it is an enzyme, then the small molecule is transformed into a product, some other metabolite, and if it is a if this protein is not an enzyme, but a receptor, then the small molecule binds and creates some signal down inside the cell, that means the cell cytosol. So, other molecules are told about this binding of the small molecule, and then according to that they have to act. Until the small molecule binds, there was no such signal. So, this is very important in many of these metabolic processes. This is what happens, that a receptor is there and the small molecule binds to the receptor. So, there are different in DNA also, if you want to have a design an anti-cancer agent, which so you have to design a molecule which destroys or breaks the DNA molecule, but to make it very specific, it should have some binding partner. The small molecule should have some binding partner where which allows it to sit onto the or to interact with the DNA and not with other macromolecules. So, basically if you have a DNA molecule, then what I am saying, that if you have a small molecule here and you want to be very specific to the DNA, so what you need is to attach something which binds to the DNA, and this goes and cuts the DNA or destroys the DNA. Okay. So, binding, selective binding is always important in drug discovery. Now, the question is, what are these binding interactions? It is like very similar to enzyme chemistry. The binding interactions could be are basically many weak interactions like electrostatic, ionic interactions, hydrogen bonds, van der Waals interactions, dipole-dipole interactions, and hydrophobic interactions. On the other hand, there could be some inhibitors or some molecules which goes and binds to the protein and then forms a covalent bond. They are called irreversible systems, okay, irreversible systems, and these are reversible ones. Okay. Now, this is the diagram which suppose this is the target, the molecular target, and it has got a pocket. That pocket is called the binding site, and now you give a chemical which is a drug and which is specific for binding to this pocket. So, that goes and binds. Now, usually what happens, there is, so basically this geometry is complementary to the binding site, but the binding site may be slightly distorted. It is not that it is totally tailor made, that it goes and binds and there is no adjustment on this binding pocket. Usually what happens, that the binding pocket is very close to the geometry of the drug, but as it goes, it changes the conformation a little bit, so as to adjust the drug from sitting properly at the binding site. This is what is called induced fit. You can see that this is slightly slanted, this binding site is still little bit wider, but as it binds, and so these two wide portions, they close the gap between this end and that end, so that the drug sits in the binding site properly. Now, this is the enlarged picture, that means you now have all these weak interactions, because why this drug will go, only geometric complementarity is not the only factor, that it goes and binds, there must be some other electronic forces, like as I told, the weak interactions. Now, as soon as it binds, if it is a receptor, then you have these signals, okay, that we know. Now, this what happens, suppose I have a receptor, where which has a natural compound, natural metabolite, and when it sits into the active site, then I develop all these signals, and these signals are transmitted by a process called signal transduction, and that creates lot of ion movements, channel opening. Basically, it starts a cascade of reactions involving several metabolic pathways, but those metabolic pathways are very important. A very simple example, if I take a neurotransmitter say dopamine, and so dopamine will have a binding site on a receptor. So, dopamine must be having some receptor, and when the dopamine sits onto the binding pocket, it creates a signal, and the overall effect we know that it creates something which elevates your mood. So, elevates your mood. Now, if somebody is very depressed something, so what you have to do? You have to make a molecule, that means his dopamine level has a problem, the concentration of dopamine has a problem. So, he has to he cannot produce dopamine because it is inbuilt in his metabolic system. So, what you have to do? You have to give a molecule from outside which looks like dopamine. So, in addition to dopamine, you have another molecule which when sits onto this binding pocket or binds to this site, it also creates signals. Okay. So, based on this principle that the drug goes into the binding pocket and creates a signal, we are talking about the receptor chemistry, we are not talking about any chemical reaction. Okay. So, here there are several things that can happen, that I can make a molecule which also like the natural metabolite like dopamine goes and binds and gives similar kind of signal. That is called an agonist. An agonist is a molecule which goes into which interacts with the binding site like the natural metabolite and produce the same kind of signal. Now, if it produces similar extent, because it is a question of that what is the extent, what is the strength of this signal, because sometimes the strength of the signal may be very high like the original metabolite, sometimes the signal may be little less. So, you have full agonist and you have partial agonist. What is full agonist? That means, if I have a graph like this, suppose the effect is something like this is a positive effect. That means, if it is the natural ligand, I put in different concentration, suppose this is concentration and this is effect. And what happens, as this is first the natural curve, that means, I do not have the drug, I add the small molecule like dopamine, the natural metabolite, and I see what is the response, what is the effect. Suppose this is the curve, and then I replace this drug and add the foreign molecule as the drug, and if it produces similar kind of effect, then it will be, suppose it produces similar kind of effect. So, it is a full agonist. Okay. But if it produces effect like this at similar concentration, that is a partial agonist. But there are some molecules, what it does, it goes and sits into the binding site and does not produce any effect. That means, it does not produce any effect. So, the natural metabolite needs more concentration. Okay. So, more concentration to stop the external molecule from binding. It is like your competitive inhibition, very similar to that. Okay. So, there are molecules, if you want to stop suppose if you have a very highly active dopamine level. So, what you need to do, you have to calm down, you have to you have to now use some molecule which sits here. So, the drug cannot bind at this pocket and it does not produce any signal or any it does not produce any signal for signal transduction. So, what will happen now? So, when that foreign molecule sits here, basically it stops the metabolic process that you are interested in. So, that will be called an agonist. So, the difference between agonist and antagonist, that is called antagonist, sorry, I am very sorry, that is what is called antagonist, sorry for that, that is antagonist. So, antagonist is basically it goes and binds and does not produce any signal. Partial agonist is produces partial signal like the original metabolite, which generally we called a ligand, because this is nothing but a ligand and a macromolecule, that interaction we are talking about. Okay. And there are some which produces very similar effect like the natural ligand, and then there are some these actually produce opposite effect and that is called inverse agonist. Now, what is this opposite effect? Opposite effect means some neurotransmitters suppose are excitatory neurotransmitters, that means they excite the system. Okay. But suppose your molecule that you are adding, that is now acting as an inhibitory neurotransmitter like system. That means, you are targeting an excitatory neurotransmitter and you are inhibiting the effect of that excitatory neurotransmitter. So, that is an opposite effect then. So, if you have inhibitory, suppose it is excitatory neurotransmitter that is having this type of curve, and if you have a molecule which produces a inhibitory curve like this, then that is called inverse agonist. So, these molecules, these terminologies are very important in drug design, and then agonists have full and partial. Okay. These are some of these interactions. I think this is already known that this is the electrostatic interactions different, and this is the hydrogen bonds, that type of interaction. It could be drug could be donor, this could be acceptor, or drug could be acceptor and this could be donor. So, lot of these things can happen, and hydrophobic is basically there are a lot of hydrophobic amino acid residues here, and the drug also has lot of hydrophobic residues in it. So, that can have this hydrophobic interactions. How the drugs are classified? Very quick, that classification of drugs is little difficult. Drugs can be classified depending on the biological and pharmacological effect that they have. Like we can call some drugs analgesics, that means pain reliever, some drugs antipsychotics, that means they affect the brain and then the mental condition, and then antihypertensive, that means they affect the blood pressure or lower the blood pressure. Then you have anti-asthmatics, so that creates that works against asthma, and then you have antibiotics that works against foreign this bacteria. Okay. However, this does not, to an organic chemist, the problem is this does not give you any structural information. Like many antibiotics are there which can vary in structure, they vary widely in structure. Okay. So, this classification is good for the doctors, that he has a list of antipsychotics, he has a list of antihypertensives, but as an organic chemist trying to develop new drugs, he wants to know, he or she wants to know what are the structures, what is the similarity between the structural similarity between these classes which are grouped together like analgesics, what are the similarity. Now, by chemical structure, that is what I said, that we are more interested in skeleton, in the organic skeleton that it has. Like we have now some molecules, some group of drugs are called penicillins, some are called barbiturates. You know what are barbiturates are also a heterocyclic framework obtained from urea and malonic acid, that gives barbiturates. Opiates, that is from the opium, and then steroids, then you have catecholamines. So, that gives the structural pattern. Catecholamines, in some cases this is useful classification as the biological activity and mechanism action expected to be same for the similar structures involved. So, for organic chemist I will say that this type of classification will be more important or more interesting to them or more useful to them. But there are other ways of classifying drugs. So, one was earlier from the biological effect, the second one from structural variation, and the third one according to the target that they interact. So, I said that every drug has a target. So, from the target you can classify also drug. Anticholinergics, because acetylcholine is another neurotransmitter. So, if you are developing agonist, antagonist, inverse agonist, all these against that acetylcholine esterase receptors. So, those compounds will be called anticholinesterases. Okay. So, they are drugs, here it is basically inhibiting, there could be two types of anticholinesterase. One is where is the receptor, you can have molecules which interact with the receptor, but another is there is an enzyme which is called acetylcholinesterase, which hydrolyzes acetylcholine. That means, which acetylcholine is a molecule which is like this. There are 3 methyls, the nitrogen is plus and then O, very simple molecule, but it is a neurotransmitter, and it is, if you hydrolyze this acetate molecule, it loses its neurotransmitter activity. So, you can have, if somebody is having low concentration of acetylcholine, then low concentration of acetylcholine, that means its hydrolysis is very fast. So, you can then inhibit that enzyme which is called acetylcholinesterase inhibition. So, the molecules which actually work on this acetylcholinesterase, remember acetylcholine works by two principles. One is, or you can control the concentration of acetylcholine or the effect of acetylcholine by targeting the receptor or by targeting the hydrolysis. Okay. Here we are talking about acetylcholinesterase, that means we are talking about the enzymatic version. So, if a molecule stops this hydrolysis, then those molecules are called acetylcholinesterases. So, this is target based. Similarly, however, again if you have target based, then you might think that only similar molecules go to the same target. So, you can have a notion that all these acetylcholinesterase inhibitors are having very similar structure. That may be true, but this is not an inviolable assumption. It says that you have to be careful. There are various dimensions of an interaction of a molecule with a target. So, if somebody is suppose there are 10 possible interactions that a ligand have with the target. Now, some molecules can utilize 5 of these, another molecule utilizes 5 of the remaining, and some molecules may be utilizing all the 10 interactions that are happening. So, you have different structural features in your inhibitor. Now, what is the drug discovery process? Let me tell you a little bit about that before we go on to the actual topic. Drug discovery process initially earlier days was kind of what is called serendipitous discovery. Serendipitous discovery means you are trying to do something and you get something else, and that is called serendipity. Okay. Like in many cases, or some drugs are discovered by looking at nature's signature. Nature's signature means, I give you an example like salicylic acid or acetyl salicylate, which is aspirin. The discovery of aspirin was like this, that there is this willow tree from which cricket bats are made. The willow tree grows near the lakes and the ponds. near the lakes and the ponds. And it may sound very strange, but people those days, earlier days, we are talking about the 17th century or 18th century. What happens, people where the not much development has happened in chemistry, or forget about biology and medicinal chemistry. So, people only look at the nature. So, they found that willow tree, which was growing in a very wet land, because it is very adjacent to a pond or a lake. So, this willow tree must have some resistance against common cold or fever, because we know that if we are drenched in a rain for a particular day, and then possibly you will get some fever or headache, or all these things can happen. So, it is very strange that people thought that willow tree must be having some compound which is making the willow tree stand upright without having any problem. Okay. So, they are actually basically connecting the tree with the human being, but anyway ultimately what they did, they took the bark and then extracted it, with water, and they started to drink this water, and indeed that was reducing the fever, that was reducing the fever and other headache, all these things, associated headache, etc. Then some, but there should be some rationale why it is affecting such things. So, people started, some chemist, buyer, they started looking at the what is the chemical that is there in willow tree, and they found that that is nothing but a glycoside of salicyl alcohol. Okay. Salicyl alcohol is nothing but this which. Okay. So, it is a glycoside, it is a O glycoside of salicyl alcohol, and then there are intelligent people, they immediately thought that this must be the compound, the glycoside must be hydrolyzing, and this must be the compound which is the active compound. And then while doing so, they found that actually this is the one that is called salicylic acid. So, they thought that salicylic acid could be a good target or to a good chemical, which may be acting on the body to have this analgesic activities. Okay. But salicylic acid is extremely toxic. You cannot take salicylic acid inside your stomach. What you can do, you can take salicyl alcohol, and that goes in the body, and then that will be oxidized by enzymes to salicylic acid, and then that can show the effect. But then what Bayer thought that this OCOCH3 made this aspirin. This is what is aspirin. So, they made the acid is there, but they reduce the acidity of this molecule by blocking it with acetyl group. So, in the body that goes and hydrolyzed to salicylic acid, and that is why this was giving the analgesic activity. Okay. But these are, you see, these are from signatures of nature, that some of these, there are many signatures in and around. So, people were looking at how nature deals with, how the other living organisms, even if it is a plant, how are they surviving with the harsh conditions. Okay. Another example, this is serendipity. Serendipity is what, that you are looking for something, you get something else. Okay. One of the classic example is penicillin, that Alexander Fleming was looking for something else, he was working with lysozyme, but in the process he discovered penicillin. But we are not going to, we will discuss that one once the antibiotics, we cover the antibiotics. Today you cannot do this you cannot wait for some serendipitous discovery to happen, or you cannot wait for somebody who looks at the nature and get some idea that we have to do this or do that. Okay. So, today it has to be because the chemistry and biology has advanced so much. So, today's drug discovery is called rational, as rational as possible. Okay. So, this is the process of today's drug discovery. First, you have to suppose somebody is suffering from a disease. So, you have to identify what is the cause of this disease. Okay. So, you have to, that means, if you can identify the target, if you can identify the cause, that it is basically some disease, okay, which is associated with high cholesterol level. Again I bring cholesterol. So, you have the biosynthetic machinery which makes cholesterol, that becomes a target. So, once you have a target, now cholesterol targets are well known, but if it is an unknown disease, or if it is a disease which where there are many targets possible, then if you identify a target, new target today, that for cancer this is the target, that this gene is the target, but you have to prove that, that is what is called target validation. If you first identify the target, then you have to prove to the community that yes, this is the target and this is the causative agent for this disease. If it is a gene, what you have to do? You have to make a system where that gene is not present, and you show that this is not suffering from the disease. Okay. So, you have to do these mutation studies. If it is a protein, you do some, again you do some engineering studies, either you delete the gene for corresponding to the protein, and you show that okay, as this protein is not there, so it is not having the disease. So, target identification and target validation is the important one. Then once you have the target, suppose it is a protein, then look for what type of protein is this. Is it a receptor? Is it an enzyme? If it is an enzyme, then try to find out what is the substrate, and if the enzyme crystal structure is known, because it is a protein, you can get a crystal structure. Then if the crystal structure is known, then what you do, suppose this is the ultimate shape of the protein, and you try to find out what is the active site of the protein. If the crystal structure is known, then the problem becomes little bit easier, because then you can design some small molecules, and then based on the active site geometry, you can design small molecules and see how it is targeting, how it is interacting with the protein. If the protein is not known, or if its crystal structure is not available, then today what they do, people do that they try to see what is the closest analog of this protein which is available in the whose crystal structure is available. That is called homology modeling. Suppose I have a protein A and the crystal structure of protein B is available. Suppose A has very similar amino acid sequence with B. Suppose 70% amino acid sequence are similar, then we call it has got 70% homology. Then as an approximation, I can take the structure of B and do my screening of the small molecules. This is nothing but what is also called in silico screening. That means, this is you are not making the molecules yet, you have a library of compounds, you write those structures, and then through computational means you try to select these small molecules. Okay. Whatever is giving very good binding, you select those molecules and then you synthesize. Now, the organic chemists come into play. You synthesize those molecules and see whether they are really interacting with the target molecule or not. Sometimes, because this computational modeling is still not perfect, so sometimes it happens that out of 10 molecules that you synthesize, maybe nothing is interacting very well. So, you have to go back and then make new sets of molecules and then again see whether they interact with your target compound. And then if suppose 1 or 2 compounds interacted well, so you have to take those molecules, those are the compounds which are called hits. So, initially the molecules from the library of compounds from in silico screening, that is computational studies, and then synthesizing and doing the actual whether it is binding to the pocket, that of course, now the question is how do you assay that how it is binding experimentally. So, that is what is called a bioassay, an assay system. Assay system means which assays the affinity for the external molecule with the to the target. So, that is another aspect of biochemistry, that you have to design an assay. If you do not have an assay, you cannot proceed. So, some assay has to be there, which will tell you about the efficiency. And then, so from heat you again sit together, and then there are many tools are there by which you can see the drug like property of the molecule. That means, some molecules which are, if it is the molecular weight is more than 1000, you can immediately say that this is not going to be a drug, although it is interacting with your interacting well with the target site, but it cannot be a drug, because for a molecule to be drug, and if it is orally taken, there are many processes that it has to be absorbed properly, it should go to the bloodstream and then distributed, and before the metabolism, before the metabolic degradation is happening, it should exert its effect. So, lot of ifs and buts are there. Even if you have a lead, if you have a heat, you have to from heat you have to select the leads which are basically you modify the heat structure and then try to come out with a better one. And once you have the lead selection, then something called lead optimization, and for lead optimization you have some tools to optimize the leads. You can use in silico screening, but there are some empirical rules I will tell you. And then once you are through these, that okay, I have 10 molecules which have got very good, at least in this is what is called in vitro. In vitro means you have the either the receptor or the enzyme, and in vitro means in test tube you are doing all these experiments, okay, or in computer. Okay. These are in vitro studies, and up to that point is you have certain number of molecules in your hand. Then what you have to do? Then you have to do what is called preclinical development. Preclinical means you have to first study how the drug is absorbed, how the drug is distributed inside the body, how the drug is metabolized in the body, and how the drug is excreted from the body. This is what is called ADME studies, ADME studies. Okay. But that means what the body is doing to the external this drug, the external chemical which is acting as a drug, and this is in general that is called pharmacokinetic studies. Pharmacokinetics means what the body does to the drug, and you have another thing which is called pharmacodynamics. This means what the drug does to the body. Okay. So, these studies have to be made, that how the drug is absorbed, how the drug is distributed, how it is metabolized, whether the metabolism is very rapid, then it will not show its activity, and then how it is excreted. If it is excreted very rapidly, then also it does not serve the purpose. You have to have some optimum concentration of the drug with a sufficient half life in the bloodstream. Okay. So, that is called PK, pharmacokinetic studies, and there is something which is called pharmacodynamics. And then another important thing is what is called toxicological effects. Toxicological effects. That means, what are the harmful effects of the drugs? Is it affecting the kidney? Is it affecting the liver? Is it affecting the heart? So, these type of toxicological effects also have to be studied. So, once you have satisfactory data here that it is not very toxic, it has got a very good therapeutic window or therapeutic, I gave you therapeutic index, and then it has good ADME properties, and it is also targeting to the intended target, that is what is falls under the preclinical development, and after that there is this clinical development. That means this part is human, you actually do it on human, and this part is partly in vivo, partly in vivo, and mainly the animal studies. Here you unfortunately have to sacrifice animals here. You have to do this preclinical studies to show the ADME properties and to show the pharmacodynamic properties and to show the toxicological effects. Once they are satisfactory, then only it is given to the humans, but that is also stage by stage. You cannot give it to the a large number of human population, okay, without knowing, because after all animal models are animal models, they cannot really mimic the human body. So, you have to be very careful. So, this is the drug discovery process. There are clinical phases, phase 1, phase 2, phase 3. If time permits, we will say what are these phase 1, phase 2, phase 3. But after phase 3 is done, then everything, if everything is satisfactory, the drug is approved, and the time taken from here to there is about 14 years. Okay. So, you see it is a very expensive and very laborious process. So, only pharmaceutical companies who are very wealthy, they can only invest money and wait for 14 years for a drug to be obtained. Okay. So, I think we will stop here and we will go to the next session and then talk about some of these targets and other related things about drug development. Thank you.